So it is really a double pleasure today for me to introduce Lucy, not only because she is a great scientist, but also she's a friend. I met Lucy when we were both postdocs at the IAS and became friends there. Lucy is a member of the brain team at Google Research and a faculty member in chemistry at the University of Cambridge. Her primary interests are in the application of machine learning approaches to better understand the relationship between the sequence and function of biological macromolecules. Before moving to Cambridge, Lucy received her PhD from Harvard University, held a fellowship in Cambridge, and was a member at the Institute for Advanced Study in Princeton. And in 2018, Lucy was appointed a Simons investigator in mathematical modeling of living systems. And today she will be telling us about data-driven machine learning models for proteins. Thank you very much, and join me in welcoming Lucy.
Thanks, Cora, for the introduction and also for the invitation. It's really nice to be here today. So I'm more than happy to take questions throughout this talk. The material I talk about will have quite a bit of biology. I don't know how much background the audience has in that subject. So if things are like confusing, I don't want people to kind of, it's sort of a Friday afternoon, so people might go to sleep, but like, please just ask a question, okay? It would be much easier. I'll also try and figure out if there are questions on Zoom, but I'm probably going to do a less good job of that. But yeah, please feel free to interrupt. So I'm also going to talk about quite a wide range of work. If there are bits that you think are sort of boring, don't tune out for the whole of the rest, because there might be something different later that is interesting. So I'm really fascinated by proteins. I've been fascinated by proteins for a really long time. Here are some basic facts about protein. As Cora mentioned, I've spent some time at Google, and that has taught me that like not everyone knows everything about proteins. And so that's why I have this slide. Proteins are made of strings of amino acids. They're specified as linear sequences. They then fold up into intricate three-dimensional structures most of the time, although not always. Sometimes they're sort of floppy and change structure a lot. So there are different types of proteins. And then they have specific functions. So here I'm showing green jellyfish. Jellyfish are glowing green because they have a lot of a protein, which is called green fluorescent protein. That's the structure of GFP above. From that structure, you can't actually tell that that protein is going to be green. You can also have cyan fluorescent proteins, red fluorescent proteins, all sorts of colors. You also can't tell by looking at the structure how intense the fluorescence will be. So structure and function for proteins are sort of closely related, but they're not they don't contain, there's information in structure that isn't in function and vice versa. So they're not totally equivalent. And that's what I'm trying to say. So there are 20 common amino acids that are most commonly used. There are also some rare amino acids that happen in different species, and they carry out complex functions. There's a lot of progress that's been made in simulation-based approaches to modeling proteins, but nonetheless, our understanding of the relationship between sequence and function is really still in its infancy. So there's a lot of progress that still needs to be made. There is a lot of data. So we have sequences for something like, at this point, 2.8 billion sequences, 2.8 billion proteins. The rate at which we're acquiring that data has continued to grow rapidly and shows no real sign of slowing down. Essentially, nature does countless experiments by making mutations and sort of trying stuff out. And then selection has the sort of ability over time to weed out those proteins that don't work so well at an organismal level. And so we have access to the record of that. We have access to lots and lots and lots of sequences, but that doesn't mean we actually know, which mutations worked well, which ones didn't. We sort of have to try and infer from the data what's going on. And there has been a revolution in our ability to both read and write DNA. So we can read sequences very cheaply and in great numbers. We can also synthesize DNA very accurately, so we can make sequences. And that way, if you train a model and get it to design sequences, you can actually make them and test them. And so, we have this huge source of information if we can only figure out how to decode it. And so biologists have been thinking about this for a really long time, right? So, even 50 years ago, when the first protein structures were being solved, biologists knew that there were differences that occurred between the sequences of the same protein in different species. And they sort of had this idea that if they looked at these differences, it could help them understand how the proteins work. And in particular, they often see what's called a compensatory mutation. So you have a wild type protein that perhaps has some pair of amino acids. In this case, I've drawn a cartoon that shows them as forming a charge pair. So there's a lysine there, which is indicated by the K, which has a positive charge, and an aspartic acid shown by a D, which has a negative charge, and it makes a nice interaction that holds the protein together, perhaps. This is just a cartoon. But then you have a mutation that happens, right? So you get this intermediate state, which is inactive. It doesn't work anymore because you've lost that charge pairing. And it's luckily another mutation comes along and restores that pairing. And that's called a compensatory mutation. So it's a mutation at a second site that's far apart in the sequence, but happen to restore the activity. And so that means that if you look at the sequence record, you never see this intermediate state because it doesn't work, right? So you only see either the wild type, the D and K, or the R and D, which is a functional double mutant. Okay, so I'm literally like looking at the amino acids in the columns of the sequence alignment. So you can see that there's a correlation there that happens because of that interaction. So all the way from sort of the 70s, people spent a lot of time trying to figure out how to look at these correlations because they felt we had lots of sequences. We can only look at the correlations and understand them, and that will tell us information about the protein. And so this is just a few of the papers. There are like hundreds of papers about this topic. And those correlations really exist. I just took a selection of hemoglobin sequences, and you can see right away that if you sort of look very carefully at the amino acids, you can pick out pairs of columns where there's actually almost a perfect correlation. So it really is there in the data. Like the reason people pick this up so early is that it's sort of a very strong signal. Of course, what you want to do with these correlations is look at sequences and measure correlations and use them to make predictions about the structure and function of the protein. And in particular, people focused on wanting to be able to predict the 3D structure because that seemed like sort of the most obvious source of a potential correlation. And so it turned out, like a lot of people worked on this, and it turned out you had to use a particular trick to get this to work, which is that you had to kind of build a global model that took into account all of the interactions across the molecule, rather than just looking at local correlations using an approach like mutual information. And so this is work that I was involved with about a decade ago. Lots of other people were involved with this too. But basically, it meant that you could go from a large sequence alignment to a set of predicted contacts in the protein, which was very surprising. And it was surprising because actually it turns out that that set of predicted contacts is basically enough information to fold up the protein. It's not perfect, but it gets you pretty close to the structure. And so here is a slide that shows some of those predicted structures against solved crystal structures. And these RMSDs are much higher than the ones that you'll see from an approach like AlphaFold. This is a completely unsupervised approach, right? There's no crystal structure information that's gone into this. You're just taking alignments, computing correlations, and using that information to fit a POTS model, and then using the coefficients of the POTS model as predicted contacts. So it was sort of very surprising that this would work. Yeah. So this is about a decade before AlphaFold. I think the advantage was that AlphaFold didn't exist. So yeah, Tess.
So how would you, so given your coefficients and being able to,
yeah.
So is it okay if I can't hear myself? That's working. Okay, great. Sorry. Okay, so your POTS model is giving you sort of like an energy function for a given structure. How would you sample the structures? Would you be using MD? Would you do?
Yeah, so actually it's even more moronically simple than that. So your POTS model gives you these EIJ coefficients. So you have an EIJ value for every pair of positions in the sequence, and you literally just take the highest scoring positions and predict that they're in contact in the structure.
I see. So you'd have to just make it.
Yeah, and the way that you fold it up is using actually an approach that was developed for NMR. So NMR gives you distance constraints between pairs of amino acids, and there's an approach called distance geometry that basically creates an embedding that sort of embeds those constraints in 3D space.
That seems non-trivial to me to actually just go from constraints to an embedding in previous ways. So yeah,
it is not. I think it is non-trivial. So this is some work that Havel and Crippen did in the early 80s. And you're right, there are actually some interest, it's a really nice theoretical result that they have. And you know, it's a wonderful algorithm.
But at this point, you can just plug in your constraints.
Yeah.
That's great.
Yeah, I didn't have to solve that. And it turns out it's surprisingly robust. So you might ask, is it better if you know distances or just sort of binary contact information, which ones are like closer than 8 angstroms or not? And that binary information is actually pretty good. It gets you pretty close to the structure. Yeah. But of course, I just told you that, you know, we fit a Potts model from the data and take the coefficients of that Potts model. It turns out that there are lots of different ways you can go about fitting a Potts model. And the approach that we used when we did that work was a mean field approach. So we made this mean field assumption, which sort of says that the correlations are small. Basically, the second order terms are small. But actually, it's the second order terms that we're sort of really interested in. So that's a little bit confusing. And it turns out if you go to the next order, so you use a message parting approach to fit the Potts model rather than that mean field approach, your result doesn't get better. And that's also surprising because if you do a better job of the fitting, if you take those sort of second order effects into account, you'd expect that it would get better. And so I was sort of always confused by this. And there is this confounding structure in the data, in the sequence data that we're using, which is caused by phylogeny. So, you know, sequences have evolved, they're not IID samples, they sort of have this shared evolutionary history. And that means that there's all this structure in the data. And so we sort of need to worry about that structure when we're fitting the Potts model, because of course this also causes correlations, right? So far I've talked about correlations between columns, so between positions in the alignment, but the phylogeny is causing correlations between rows because some of the sequences are very, very similar to each other. So now we have two different sources of correlation, right? We have column correlation and row correlation. And if we think about a covariance matrix, right? If you take the row covariance matrix and the column covariance matrix, they have the same information in them, right? There's only one set of sequences, and so there's sort of morally only one covariance matrix, right? So basically, and so these two sources of covariance or correlation are getting kind of convolved with each other, and we need to worry about how to pull them apart or deconvolve them. Does that make sense? So you sort of thought about this, and maybe the simplest thing to do is to make a very simple simulation, right? If I have a protein, this is a contact map. I think this is DHFR, it's dihydrofolate reductase. I can, you know, take the sequence and simulate evolution along a tree, or I can simulate evolution without a tree. So having a tree just means that every so often, if I start with a sequence, I sort of add mutations, and I move along the branch, and then I duplicate. And then I have two sequences, and yeah.
If I get it correctly, the tree represents evolution, and is that between species or within one species?
Oh, between species. It could also be, you could also have strains within a species. So I think that this is just a sort of abstract representation. Yeah. But essentially, you know, with phylogeny, you're going to get more of a correlation structure than without phylogeny. And so it turns out if you do that and then carry out this inference procedure, if you didn't have any phylogeny, you'd be able to find the contacts perfectly. But the phylogeny kind of causes a lot of confounding information, right? So this is, when I say find the contacts perfectly, this is just by looking at the covariance. So without having to fit the Potts model, just by looking at the data and taking the covariance. So I'm sort of now wondering, this whole sort of Potts model procedure only seems necessary in this case when you have phylogeny. If you don't have phylogeny, you could just look at covariance. And so maybe what this Potts model approximation is, or Potts model sort of formalism or explanation is really doing, is providing a mechanism for getting rid of the phylogenetic information. And so we thought a little bit more about phylogeny, and we realized that we could actually write down a model that captured phylogeny quite accurately. So if you have phylogeny, you're going to end up with a matrix that has sort of a covariance matrix that has off-diagonal entries. And because of the branching structure, those off-diagonal entries will have essentially a particular form. So here we defined a parameter alpha, which basically tells us something about the length of the branch compared to the length of the sequence. And so you can imagine phylogenies have all sorts of different shapes, but it turns out you can just consider an average number of mutations per branch and define this parameter. So these matrices have a very nice sort of regular form. And it turns out that their eigenvalues have, which I've shown here, lambda, follow a really nice power law. So lambda goes like r to the minus beta, where beta is a parameter defined from alpha. And so this tells us that if we look at sequence alignments, compute their covariance matrices, we should see a power law in the eigenvalues. And it turns out actually that's true. This was like really remarkable. It's an alignment of 30,000 serine protease sequences that we downloaded. If we compute the eigenvalues of that matrix, we find that they lie on a power law exactly as the theory predicts. And so, you know, up to this point, we sort of thought that we were doing all this nice theory and it was kind of fun, but, you know, I don't know, like theory doesn't often correspond to reality. And I didn't actually expect this to happen when we computed the eigenvalues of the covariance matrix of the alignment, but it did. And so then we could ask, if we take that covariance matrix and we consider different truncations of the outer product expansion of that matrix, and we use those different truncations to predict structure, we can ask where in the spectrum, in the set of eigenvalues, where is the information that tells us the most about structure, tells us the most about being able to predict contacts. And it turns out that it's in the very small eigenvalues. These large eigenvalues are dominated by phylogeny. And you can kind of see that because that power law is sort of holds for the largest eigenvalues and only deviates when you get to the very small eigenvalues. And so it turns out that if we just consider the small eigenvalues, we can do a good job of predicting contact maps from sequence alignments. And if you take that mean field approach to fit the Potts models, you basically end up inverting the covariance matrix. That's how you fit the Potts model. When you invert the covariance matrix, of course, you basically send all the eigenvalues to lambda to 1 over lambda. You're massively upweighting the small eigenvalues. And so what I think is happening is that that sort of Potts model explanation or approach to fitting the covariance is really just correcting for or removing the phylogeny, which dominates the large eigenvalues. That's what I think is going on. Yeah.
So I have a question that I think will relate to this slide, but it goes back to when you're computing the covariance with and without phylogeny. Is it correct that in the case with phylogeny, you're kind of computing, like, let's say we have, 10 sequences that are 50 characters long, I'm kind of like flattening it and then computing the covariance of every entry to every other entry, and then maybe reshaping things and then summing over certain axes. Versus in the case where I'm not caring about phylogeny, I compute kind of like the outer product of each sequence with itself and then just kind of averaging, but not assuming any correlation between sequences. Is that? So
you don't make any, when you compute the covariance, you do it exactly the same in the two ways. So in both ways, with both simulations, you end up with a large set of sequences. It's just that one kind of is made, using a phylogenetic tree, and the other is made just letting them all evolve completely independently from each other.
So it's just which sequences you're.
Yeah. So in both cases, yeah, you end up with a large sequence alignment, and which sequences are in that alignment is sort of dictated by whether you had phylogeny or not. Does that make sense? And so in one case, you have basically not exactly IID sequences, but sequences that are sort of are less constrained because there's no phylogeny. And in the phylogeny case, because you have this sort of duplication that happens every so often, the sequences are more related to each other. If that makes sense. Yeah, sorry, there's like a lot of ideas that I'm trying to talk about very quickly.
Thank you.
Thanks. Yeah.
So if you were to like truncate the SVD style, but kind of backwards and get the small eigenvalues instead of the large ones, would you do better? I understand that right?
Yeah, so this is exactly, so sorry, I did also explain this to you quickly. There's like a lot of eigenvalues in this matrix because it's the length of the sequence times 20. So you have 20 possible amino acids at each site. So that's why this is like over 4,000, there's over 4,000 eigenvalues. And what I'm doing here is I'm taking every single possible truncation of that outer product expansion. And I'm sort of taking just the smallest and the next. So all of these possible.
In reverse though, because you're starting with the smallest ones are the ones that matter.
Yeah. Yeah, so you're right, it is in reverse. So I could have made the plot kind of the other way around, starting with just the largest one and kind of going. Yeah. Yeah, exactly. Yeah. Yeah, and it's because basically all of these ones here, so this is the eigenvalues in rank order. This is like the biggest one to the smallest one. And all of these large ones really fall on this power law, and so they're completely dominated by phylogeny. And so in the case where there's no phylogeny, we don't see that. We see like the spectrum doesn't have that power law. It just it actually flattens. It turns over and yeah. Yes.
So I'm just wondering, the alphas that are your off diagonals in the covariance matrix, which I guess related to the exponent there in your power law, why is that the same in every branching?
I'm showing you just like very regular trees here. Do you mean like if the tree isn't regular?
Yeah, I mean, so then on the next slide, it looks so nice in power law, and I sort of would have thought that you'd have different alphas and this would get messy, but
yeah, it actually doesn't. So I had a really great graduate student who was very mathematical, and she went through and proved that if you took alpha as the average of the branch length across the trees, as long as I think the fourth moment of the branch length had to be bounded, there was some condition that had to be true. As long as that was true, then this actually holds for trees no matter kind of what the structure is. You can actually have trees where branches are missing, and this still holds. So I haven't put all the sort of details, but yeah, there's actually a really elegant theory. Grad students now at DeepMind. So yeah, and so I guess what am I hoping? So this is the picture from the AlphaFold paper, which I think this is like, recently come out. This is AlphaFold 2. And I think what's interesting here is that we see exactly the same kind of use of a sequence alignment as input, and also this sort of pairwise representation, and an awful lot of supervision. So you can see that there's like structural templates being used from the start. And of course, during training, the actual 3D structure of the protein is available. And so what I kind of think is happening here is that essentially the same information is being used, right? Like we have this sequence alignment we know is sort of really important. I think there are cases where there's a very, very small alignment where this AlphaFold doesn't work nearly as well. But because we're supervising and there's all this information about structure that's being put into the model, you're able to do much, much better, right? So the structures that come out of AlphaFold are really great. But I think what I'm trying to sort of get at is that covariance information, or the information in the data, is broader than structure alone. So if we just use the information in the data, we get sort of rough approximation of structure. If we supervise, we can sort of almost fine tune it or push it much towards much more closely towards structure. But I think that there's actually like a lot of other information, and perhaps even information that reflects function, that we can also extract from that data. So that's what I want to talk about in the rest of the talk. So I'm really interested in this direction, sequence to function. I think that being able to predict structure is great, but it's actually also really important to know what the proteins do. And so the rest of the talk is going to be about being able to predict function from sequence. And actually, it turns out we're really bad at this. So this is a Nature paper from 2018. And as you can see, it opens by saying that 1/3 of all protein coding genes from bacterial genomes simply can't be annotated with function. We just don't know what they do. We have all of these sequences. We spent lots of money to figure out what they are and put them in databases. And they're literally labeled uncharacterized proteins. And that's just not much use. Like if you're trying to solve a particular problem in biotechnology, having a load of uncharacterized proteins doesn't help. And so this paper was sort of a tour de force. They identified phenotypes for almost 12,000 protein coding genes, which is great, but 12,000 is kind of a small number. Yeah.
Sorry to interrupt. Could you go back one slide?
Yeah.
At least maybe it's just my poor understanding of biology, but I've always thought that structure and function are more proximally related than sequence and function. So, you would think that there's an arrow from down, from the structure to the function. And I guess I'm curious, there's graph neural network approaches and stuff like that to learn that arrow and why that's not easier. I mean, it seems like there's like a smaller space to learn from. Maybe that's just like bad intuition.
No, I don't think that's bad intuition. But I guess if we can get from sequence to function, sequence to structure, right, then it seems like we can kind of just, sort of shortcut and go straight to function. Maybe more realistically, it's probably going to be important to include structure as well, right? Like, I don't know. If all the information about structure is in sequence, then we shouldn't need to add it. On the other hand, usually adding more data helps. So I think that sort of remains to be seen.
It was like we could be thinking of filtering down the right. If I think of an enzyme, right, it's actually the shape that allows it to be the enzyme.
So it's an interesting point that in particular the shape around the active site for enzymes is really, really important. And actually, even using the unsupervised approach, we would get really high resolution just around the active site. or we'd get much lower resolution kind of in the backside of the enzyme, where the structure is sort of less important. And so I think that there is sort of an interesting gradient between cases where structure is really, really tied to function and cases where it's actually, much less, for an antibody, for example, right? They're sort of squishy and they assume a structure upon binding to their target. But
Thank you.
Is there more data on function or structure?
that is an interesting question. There's probably, I actually.
This might be historical, because it's somewhat easier to measure function. Right?
Well, that's all. There's also lots of different functions to measure. I don't know if I could even sort of make a guess at whether there's more data about structure or function. There's a lot of data about structure. There's like almost 200,000 structures in the PDB. There's also a lot of data about function. Yeah, I don't know. Yeah.
You've mentioned that it's possible that the sequence doesn't contain all the information about either structure or function. Where would that additional information be coming from in that case?
Yeah, this is a great question. So I could sort of point to perhaps chaperones that are necessary to help some proteins fold. So maybe somehow there is some information from the environment. The fact that you sort of had to add supervision for AlphaFold to do so well kind of sort of makes me wonder. It may also just be that the information is all there and we're just not doing a good job of extracting it, right? Yeah. Cool. So maybe a sort of way of stating this challenge is, can we build models that capture the relationship between protein sequence and phenotype? And I'm sort of using the word phenotype to perhaps sort of encompass both structure and function. And this is like sort of some sort of vision about what that might look like. You have sort of islands in this space, which are different proteins, and there are sort of places within each island where the protein is sort of better or worse somehow. This is from a paper from 2006. I don't have a better way of visualizing this. I have no idea what it should look like, but somehow there should be some kind of representation or embedding of sequence that allows us to sort of understand better the relationship between sequence and function. And so I think there are sort of two key challenges. There's protein discovery and protein optimization. I sort of have parts of a research program that talk about both of those. I may only have time to talk about the discovery part, but I think that there are interesting challenges in both. And essentially, if we can do a better job of modeling this relationship between sequence and function, then it will have lots of useful benefits. Like, for example, greatly reducing the amount of experimental data that we have to collect in order to understand or discover a protein with a particular new function or improve the function of a protein in a way that we need. So an example might be, if we're trying, so there's been a lot of excitement recently about enzymes that degrade plastic, in particular PET. And people have shown that you can actually change enzymes. There was a Nature paper recently showing that you could modify this particular enzyme so that it will degrade PET at room temperature in sort of a reasonable time frame. It'll actually kind of eat plastic bottles, which is kind of exciting for if you're thinking about recycling. But there are lots of other kinds of plastic that we don't have enzymes for. And so it would be really nice, perhaps, in all of those millions of uncharacterized sequences, there are some enzymes hiding out there that will degrade PLA, for example. So in terms of optimization, you can think about running an active learning loop where you synthesize specific designed sequence variants, test them in the lab, and then use that data to build or refine a model, and then optimize the model kind of in silico, and then use that model to select the most informative next batch of sequences. Of course, there's some trade-off between exploitation and exploration there, as you often see in these kind of situations. And this is something that we've been actively running with experimental collaborators. And what's nice here is that synthesizing DNA is actually relatively cheap. It's it's difficult to do experiments, so I certainly can't do them. But in contrast to small molecule chemistry, where synthesizing molecules is itself a challenge, for proteins you can literally print the DNA and then use cells, organisms, to make the proteins. So it's a much more tractable space in that sense. And so we have collaborators that have synthesized even hundreds of thousands of sequence variants for us in these active learning loops. You can collect large amounts of data. So yeah, there are a few things that I want to talk about. And I suspect I'm only going to get to talk about the first one of these, which is using deep learning models to annotate protein sequences with function more accurately than state-of-the-art methods without needing alignment. So what do I mean by that? So essentially, there are lots of existing approaches to annotate sequences with function. They often involve basically aligning a new sequence with the most similar existing sequence you can find in the database, and then just transferring the functional annotation. Right? So if a sequence is similar enough, then probably it does the same thing. Alignment is sort of fairly expensive computationally. So these methods aren't, it actually takes a reasonable amount of time to run them, especially if you're trying to do all-by-all pairwise comparisons. And as I said, we have something like 3 billion sequences in the database, so there's a lot of data. And so we're interested in asking if we can use deep learning to do a better job of this process, but also we're interested because it'll sort of allow us to learn a representation that we can then potentially use to both label new sequences or predict the function of new sequences, and also to optimize existing ones. So this is a task. It's very simple. We want to take amino acid sequences, build a model that labels them with functional annotations. And we're going to start by setting this up as a classification task. We use data from a database called PFAM, which is a protein family annotation database. And this annotates protein domains, which are sort of the core conserved part of a protein. So in a protein sequence, you tend to have sort of bits at each end that change a lot, and kind of a specific different sort of instances. And then you have the sort of core piece, and there can be more than one domain in a protein. And those pieces are relatively conserved across evolution. You see them all over, sort of in lots of different species. And so you can build models. And people for a long time have used hidden Markov models to kind of capture these functional domains. In the hidden Markov model, you can also use that to cut the sequence, so to tell you where the domain is. And so that the database PFAM has all of these annotations. So PFAM has two parts. There's PFAM4, which is about 60 million sequences, that's grown using HMMs. And there's PFAM seed, which is the sort of small, well-annotated set of sequences that's used to build the HMMs. So it's mostly human curated, and the members are chosen as kind of examples or exemplars of the families. And so we have something like 1.4 million protein sequences, and there's almost 18,000 classes. So there's not that many examples per class. And the reason that we're particularly interested in PFAM seed is because it's human curated, right? If we look at PFAM4, we're basically learning HMM annotations, which is less interesting. But if we're looking at PFAM seed, then we're learning something that involves human expertise. And so even though the full database has more information in it, we sort of spent our time looking at the seed. So this is some information about the distribution of the data set. There's a big distribution of sequence lengths. There's also very, very many small classes. So there's like thousands of classes with 25 or fewer members. Yeah.
So when you talk about the function and the family of a protein, I mean, to some approximation, every protein has its own function. And so there's as many functions as there are proteins. But presumably you're trying to not quite. So that's what I'm asking. So how broad of a level are you classifying the function? What do the families mean? What is this? What do these labels look like?
So globenfold would be a family. So the globenfold or hemoglobin, the alpha and beta chains from hemoglobin, that would be a protein family, or a serine protease, or sorry.
Those are individual proteins.
No, but there are very many versions of them from different species, right? So we have lots and lots and lots of versions or variants of each of those proteins. So I think when you think about a protein, roughly speaking, a protein will be a family, right? But because we have lots of variants, so serine protease is a good example, right? There are versions of trypsin from lots of different species that have a lot of sequence variation. There's also serine proteases with different specificities, like kallikrein or chymotrypsin or any of the other. These are all enzymes that cut peptide bonds, and they cut peptide bonds between different pairs of amino acids.
Different families can have the same function?
No, so the sequences within a family have the same function. They can have different specificities, for example, right, in the sense that they're cutting the peptide bond between different pairs of amino acids, but that's sort of, that's considered the same function, basically, or a very closely related function. And so you're right, you could sort of draw this distinction at whatever level you wanted, right? And there are indeed classifications that you look at sort of families or superfamilies or you know, sort of classes of folds. But in this case, we're talking about families basically where the sequences have the same function. And the variation is from evolution.
Another question about like functionality. Do you ever have sets of proteins where their function is very different in either different environmental settings? So where it's like, just from the sequence, I can't quite tell you exactly what it's going to do because it depends on the context in which that protein is situated, or do proteins tend to be more specialized where you can kind of say something more specific?
Yeah, that's a good question. There are certainly proteins that will denature, for example, at different temperatures.
Where it loses the function.
Yeah, exactly.
Is it ever where they like change function or?
Yeah, I can't think of examples offhand where they change function. I'm sure probably there are some examples, just because, but generally, yeah, I think generally those are not common.
Proteins tend to be very specialized.
Yes, yeah, exactly.
Am I understanding correctly that you have 1.4 million human labeled examples?
Yeah.
How did you get those?
Well, so PFAM is a database that has existed since the 1990s. Every family actually has an author, like a person, who has literally looked at the alignment of sequences contained in that family and kind of signed off on it. It is the case that some of those families have been updated because they wanted all sort of seed sequences to come from reference proteomes. So there have been some changes. But yeah, so I mean, in the same sense that, you know, all of those 200,000 structures in the PDB have been solved by individuals, right? And it takes a sort of a, you know, reasonable amount of time and effort to solve a structure. You know, it is the case that people have spent an awful lot of time looking at sequences and sequence alignments in order to kind of confirm that they're good. People are really good at this. So we actually work with Alex Bateman, who is the PI of PFAM, and he can tell you he can look at a set of sequences very quickly and kind of tell you a lot about it. So there is this sort of real expertise involved. It's not the case that every one of these sequences has been experimentally validated. So that's certainly not what I'm saying, but there is human oversight that has gone into this database.
Cool, thanks.
So we've got a pretty simple model. This is a ResNet. It uses dilated convolutions, which we found to be a really nice way of stopping the model from exploding, as the sequences can be really quite long. So you can put any length of sequence into our model. We make a 1100 length representation of each sequence. We learn that using this convolutional box. And it's not like we spent a lot of time optimizing the size of that representation. This seemed to work pretty well. We wanted our model to fit on a single GPU, and so that sort of provided the constraint. We could undoubtedly have made a better model if we hadn't implemented that constraint. And we just felt that, like, in order for it to be sort of practically useful and like people to be able to use the model, a single GPU was kind of already kind of a lot of resource, so it seemed a bit much to use more than that. This is performance. So I'm showing error as a function. I mean, of course, we want error to be low as a function of distance from the training set. So we've already talked about the fact there's a lot of structure in the data, so we have to be very careful. We made a random split. Of course, in our test set, that means that we have sequences that are very, very close to the training data set and also sequences that are far away. And being able to, you know, do a good job of predicting the label for sequences that are very close to the training data set is kind of, you know, just requires you to memorize. It isn't really impressive. But over here, you know, as we're getting further and further away from the training set, that's where we want the performance to be good. And compared to the HMM approach, which made something like 1,700 errors across our test set of 126,000 sequences, our CNN was doing better, and our ensemble of those CNNs did even better with only 200 errors. So our ensemble actually contains 19 copies of the CNN. So I guess now we're using 19 GPUs, which is kind of getting a bit ridiculous. But it does do a good job. In the field, people are very, very aware of this problem with structure in the data, and so they make what's called a clustered split. And this is because often when you want to annotate sequences that are in the database that aren't characterized, the reason you can't annotate them is because they're very far from anything you've seen before. And so to mimic that, we made a clustered split. Here, every test sequence is no more than 25% identical to any training sequence. And so we had a smaller test set because it's hard to find those examples. We split every family. And so it turns out this is much more challenging. So you can see the error rates are higher here overall, but our ensemble is still doing better. Our single model is not doing better, so it's sort of embarrassing. HMM actually beats us, but the ensemble saves us there. And it turns out we can also use the representation to do a good job of this task. And so without requiring any additional compute compared to the CNN, we can use this learned representation to classify each held out test sequence just using a very simple approach. And it turns out that sort of per family version actually reduces our overall error rate and also reduces our small error rate on small families, which we were really worried about this number. And so here I've stratified performance by training set family size. And you can see that for the very smallest families, the HMM is still doing better. But as soon as the training data gets a little bit bigger, the ensemble does best. But what was interesting is that this deep learning approach actually provided complementary information to the existing approaches. So if we ensembled the two, then we could come up with a method, very simple method that did better than either of the individual approaches. That's shown here. We get this performance improvement. If we ensemble two of the existing approaches, we get no improvement. And so this was sort of exciting enough that it convinced people at PFAN that they should trust this approach, and we added a sort of significant fraction of sequences to the database. And we're going to add some more in a couple of weeks, which is exciting. Yeah. Oh.
My question is why not?
Yeah, so then we were sort of wondering if we could do any better by pre-training. And so we use this very simple masking approach, sort of self-supervision approach to pre-training a model, and then simply retrain the last layer for classification. It's a completely standard approach, so we didn't really do anything novel here. And it turns out that this compares training from scratch with the pre-training. And you can see that in particular for the 12-layer model, it really makes quite a big difference. And so we're able to improve this. Actually, this 90.5 is better than our ensemble approach. And what's particularly exciting is that we have a lot fewer parameters. And so at inference time, it's quite a bit cheaper. This is important because we want to actually make these models available. It would be useful if people can use them without having to have enormous amounts of compute. I was very surprised here at how many parameters the existing state-of-the-art approaches have. So that was a surprise to me. Yeah, they're not lightweight in any sense. And you can see that this is now showing accuracy rather than error rate. I apologize, I flipped the axis, but the transformer approach is doing just a little bit better. We didn't actually put a huge amount of effort into really optimizing this, so maybe it could do even better. I don't know. And yeah, again, for the very small families, it's given us a bit of improvement, which was particularly exciting.
Can I clarify something?
Yeah.
So are you just predicting whether sequences are in the same family, or is there some way of actually telling what the function is?
So we're predicting which family they're in. There's 18,000 possible families, right? And we're predicting for each domain sequence, we're predicting which one it belongs to.
But not anything about the function itself. So for example, there were a bunch of proteins you mentioned that we don't know what their function is.
Yeah.
So you can't tell me what the function is of those proteins.
Well, not with this approach, but hopefully, well, actually, I'm kind of running out of time, but hopefully with the approach after this one, we'll be able to get there. Yeah, so you're saying that it's a closed domain task, right? It's sort of pointless if we have all these uncharacterized proteins that actually have new functions. And so, yeah, we're trying to solve that problem right now.
Right. And the other question was about structure. You said you don't use anything about the structure. of the protein itself, but that seems like that would be useful.
Yeah, and so this is something that we're sort of doing as we have this conversation. We're trying it out. What we've shown so far is that adding secondary structure doesn't improve things. So secondary structure is like, you can predict that pretty well from sequence. It turns out if you add that in explicitly, like we add in the secondary structure from the AlphaFold predictive models, doesn't give us any lift.
The full AlphaFold prediction might.
Yeah, absolutely. I don't know yet. It's true. Yeah.
Is there a mechanism for telling you if your sequence doesn't fit into any of the 18,000 buckets?
Yeah, that is a good question. So we sort of implemented that. We made a split where we held out all of the small families. And then sort of to make that work, basically what you have to have is you have a test sequence, and if it doesn't, you have to have a threshold, and if it doesn't clear the threshold for any of the existing families, you kind of posit that there's a new family. We haven't really kind of done that properly, but I think it will work. Sorry, it's like my unjustified claim. But what I want to sort of quickly say here is that we sort of made new models that take this a little bit further. I talked about the fact that we were using pre-cut domain sequences, which are kind of cheating. What we need to do is take the full amino acid sequence and label every residue with both whether it's in a domain or not, and if it's in a domain, which domain it's in. And so it turns out that actually works really well. It's basically exactly the same kind of model. We just sort of label every single residue rather than labeling the whole sequence. And that gives us both the domain call and which residues our model thinks are in the domain. That really works well. And so I'm actually able to use that in collaboration with Magnify. So Magnify is a metagenomic database at EBI. There's a lot of sequences in Magnify. We were able to annotate 1.5 billion of them. And in particular, we could annotate something like 200 million proteins that couldn't be annotated using PFAM and couldn't be annotated using traditional alignment-based methods. And so that for us was really exciting. It was kind of a demonstration of how the models could actually be useful. And so we will continue working with Magnify to update this. But yeah, absolutely. We have this problem that we're dealing with closed domain classification tasks, right? Which is sort of boring. We really want is models that can tell us about new functions that we haven't seen before. And so for this, we turn to some of the language models that have been recently developed. And sort of thinking about this as translating between amino acid sequences and text. And so this is an illustration of a T5 model. I've stolen this from the T5 paper. Replaced the tasks and prompts with protein-specific ones. Essentially, here we give the model a protein sequence and a prompt that asks a particular question about that sequence, and it tells us, the idea is that it tells us the answer. And so as proof of concept, we first trained these models to perform the PFAM classification task that I've talked about so much. So we first sort of dreamed up this task as kind of like the MNIST of protein function prediction. It turns out that actually having this sort of small, well-defined task is really, really useful, even though it's sort of, it's not exactly a done task, but it is not like it is kind of a bit like MNIST, I think. And so we trained this model to jointly predict any permutation of 3 different tags, one of which was a free text PFAM family description. So PFAM has a short 1-line description for every family. And so we asked the model to generate a description, also to predict the accession, which is like the sort of index, and they also have a family ID. So 2 and 3 are closed domain tasks, but the first one is a text generation task. And so the model can actually predict things that weren't seen in training, right? It can give whatever phrase it wants. And so we trained it. This is like a completely canonical approach. We just took a random subset of these 3 tags and appended them in random order between each sequence given during training. And so then at inference time, you give both which fields you want to predict and then the associated protein sequence.
I'm curious, was this a fine tuning or from scratch?
This is from scratch. We have actually also tried fine tuning in the sense of using like a pre-trained protein model, and we sort of haven't, I don't think we've done like a great job of this, but our initial sort of experiments didn't suggest that it added anything, but we should probably revisit that. Yeah. Yes. Yeah, I realize I don't have any on this slide, or the next slide. Yeah, they're like text phrases. Hang on, I think I have a spreadsheet full of them open somewhere. I can read some of them out. Yeah, so for example, A49-like RNA polymerase I associated factor, or alanine racemase N-terminal domain, or Trove domain, or carbon nitrogen hydrolase. So it's a fairly unusual sort of set of text phrases. So we also could have used a pre-trained language model, but it's not clear how much a pre-trained language model would know about these particular phrases. It is a bit sort of.
I guess I'm curious. Are all those words like it's a match? Will it mean something if the model puts together?
So this is a great question. So what we found, as this plot shows, is that it does pretty well at the closed domain task. That was good. Sort of, you know, we were relieved about that, I think is the right expression. And then, sorry, I've lost the slides. So then what we asked is, you know, how well does it do at description generation? Of course, we have to decide how to measure that, right? So the simplest way to measure it is just ask for exact match with the PFAM generated description. So then we're just saying we're sort of back to a closed domain task. Does it like perfectly, you know, regenerate the training data? So it turns out we get something like 88% accuracy if we take that exact match metric. But the whole point is that we want the model to generate new descriptions. So there's sort of a tension there. And among the errors, so there's something like 11% that are errors, we find around 270 model-generated descriptions that were never seen during training. So sometimes it's just giving you the wrong training phrase, but a little bit of the time it's actually giving you a new description, which was kind of exciting. Some of these are fragments or nonsense, right? And you can sort of tell that pretty clearly because they look like nonsense. But actually, quite a few of them are viable family descriptions. And so then we're left. Yeah, so we're sort of stuck. And the problem is that, you know, we're looking at image captioning. This is all very well, right? The model says dog catching a frisbee, and we can say, yes, it is a dog catching a frisbee, more or less. But I have this protein sequence, and the model tells me it's a capsid protein VP1, and well, I don't know, right? Like, sort of the whole problem. So we have this sort of horrible, you know, situation where we don't know how to judge the phrases that the model is giving us. And so basically, in order to try and fix that problem, we have these, you know, model generated descriptions that we think look good. We have to turn to the experts and ask for expert manual curation. And so we actually now have ended up working with Uniprot. So people had this sort of very restrictive vocabulary because there was one description for each family, and there's only 18,000 families. I mean, we only have 18,000 descriptions in our vocabulary. Sort of a bit limited. So we moved to working with a database called Uniprot, which has a name for every protein, and there's something like 230 million proteins in Uniprot. So it's kind of quite a bit larger vocabulary. And they also have a set of expert manual curators, expert curators who could help us look at the names that were generated. It's a fairly well-used database. A lot of people look at this and use it. And something like 30% of the proteins are actually uncharacterized. So they're named literally uncharacterized protein, and there isn't that much information about them. And so we made a temporal split of the data. We take the test set, we could immediately corroborate something like 57% of the predictions, but of the rest, we had to go to manual annotation to judge whether they were correct or not. We took a stratified set of 100 predictions, stratified across model confidences. We wanted to know, you know, get some idea of whether we could trust model confidence to calibrate our predictions. And basically, there were cases in which the curators preferred neural network, cases where they preferred Uniprot, cases where they only had a syntactic preference, not semantic, and then there were some cases that were errors. And so basically, it seems like the models are doing reasonably well. Sometimes they get things wrong. So, you know, they're sort of like 85% of the time we're doing okay, and sometimes the models seem to not be doing that well. So people like experts, there are people who have, you know, PhDs and who are professional curators. who work at Uniprot in order to help curate the existing names. And so they have looked carefully at our names and tried to figure out if they're correct or not. These are all already characterized proteins. They have names, right? So it is a ground truth. So the curator is just trying to say, is our predicted name sort of better, or doesn't mean the same thing? It's a synonym. When we get to the uncharacterized proteins, it's a whole different ballgame because we don't have any ground truth. And so we're trying to figure out whether those predictions are good or not. And it seems like we can actually provide names for a reasonable fraction of these uncharacterized proteins, something like 35 million proteins, where the name, it seems likely, would be preferred. And so that's something that we're working together with on Uniprot. We hope to make a release in the near future. Yeah, and so we have a lot of next steps. Like, this is like a very much sort of initial task, but I think the idea of using these models to really generate new information, even to just sort of synthesize all the information that's in the literature. We've seen examples in other areas of science, I think, where people have trained models to do this. And here it seems like we have a really rich source of data. Yeah, so thanks so much for all the questions. I think I'll stop there. And
Yeah, so say you're dealing with fluorescence, for example, right? If we want to think, okay, how can we make better GFP, right? More fluorescent GFP, that's like a number, right? So you can assign, and there's a big, there's a huge annotated 50,000, different mutations of GFP in various things, because, people do. That's not really a translation task anymore, right? It's more like a regression, or if you want to do classification or ordinal classification, maybe if you want to do buckets. So like, what would you do there? You just take the transformer embeddings and then fit a classifier on top or a regressor?
yeah, I think it's I personally think it's kind of an open question whether the transformer embeddings, like using that pre-trained model, kind of adds versus just training a model from scratch across experimental data. I'm sure it will. I just haven't sort of yet seen the demonstration that I'm completely convinced by. But yeah, that seems like it should be the right way to go. Yeah. So I have sort of the second half of this talk is all about doing exactly that kind of optimization task, but actually I haven't used any of the sort of pre-trained any of the embeddings. So I think there are sort of two sort of areas of research here that hopefully will come together, and there'll be like a beautiful synergy, but I haven't managed to make that happen yet. So yeah.
Thanks, Liz, for the great talks. So you have decisions that you make when you're designing these models, and in one of the models you talked about, you decided that you wanted to have a latent representation or an embedding space, and I think it was 1,000-dimensional space.
Yeah, 1,100. Yeah.
And can you kind of describe what was the motivation for that? Did you already anticipate that you were going to do some kind of clustering in that space to improve performance? But was it helpful to have that kind of intermediate representation prior to the final decision?
Yeah, I think that's a good question. I don't know if it was sort of helpful in terms of the initial classification task, right? Like we could have just gone end to end without needing it. It was nice to be able to sort of pull it out and ask what the model was doing, and then using that representation really did it sort of improve performance, and also sort of lent itself to this prediction of new families. So I didn't really talk about it, but we did actually sort of do this thing where we held out families and we were able to show that we could kind of recover them using that learned embedding. So I think that's actually going to become more and more useful and important as we build richer models and incorporate more sources of information. And I think being able to sort of move around in the space. So we found that we can actually move around in sequence space relatively easily, that we can make multiple mutations to a stretch of sequence and retain function or even improve it. And so I think figuring out what moves to make, if we want to do it without collecting huge amounts of experimental data for the particular functional task, it seems to me like the embedding should be a really rich source of information. There's a lot of claims being published right now that is the case. It's sort of a very attractive idea. So I think we have to be really, really careful to try and ask, when are we actually getting lift versus, when could we have done just as well without using that. So, and it can be really tricky, honestly, to tell. Yeah. Phew, maybe I've exhausted you.
Okay, I guess if there are no more questions, let's thank Lucy again for a super interesting talk.